Yeah, thank you very much, Mukund. So is this working?
Yes.
Yeah. So what I want to do today is I'm giving 3 lectures over the course of this school, and what I want to do today is talk about the evolution of HIV, and you'll see in a minute why. Because it's one of the systems where we can really observe how a population changes from 1 week, 1 month, 1 year to the next, very directly in a fairly stereotypical environment that is the human body. And then on Monday, I want to step back a little bit and explain some theoretical concepts that we've developed partly before we got into this, but anyway, it's a good motivation. Yeah, and then in the last lecture next Thursday, I want to talk about our efforts to sort of predict and track pathogens, not only HIV, mainly influenza, but also Zika and Ebola viruses and other things. So just to set things off, you might have seen this graph over the past 2 weeks, right? It's sort of an 130-year-old depiction, 140 by now, I guess, of how people thought of how evolution worked, right? That there is some hierarchical structure that sort of brings together all the different living things in one common ancestor. And this sort of illustrates one of the problems that we have studying evolution, right? Because we have the status quo, and everything that's on the leaves of this tree is now, and you're trying to work your way in, but you really don't have any dynamic information to test whether these, it's ultimately a very dynamic process, right? But without dynamic information, it's very difficult to test the theories and the models that you come up with, whether they hold any water. And I guess what has happened over the past sort of 20, 30 years is sort of an attempt to go from these sort of qualitative descriptions as evolution as a hierarchical process where things change step by step to something quantitative where we try to figure out what are the relevant parameters, sort of to what, how do the response of a population depend on different parameters, to what extent is a particular process repeatable or predictable, how gradually is it, and so forth. And one technology that has made this effort possible, or at least much more amenable over the last couple of years, is the development of high-throughput sequencing technologies. As this, I guess probably somebody had a similar graph of this sort over the past 2 years. Did somebody? So what you see here on the Y scale is the gigabases a particular sequencing machine gives you for one run, right? For some of these, the run is a day, for some of it, it's 2 weeks, but generally, it's a scale from, that's a megabase. That's a gigabase. The human genome is roughly here. So that would be 30 human genomes. And on the x-axis is on a logarithmic scale the read lengths that these instruments return. And that's in the early days, one of the earlier high-throughput sequences, Solid and the Genome Analyzer 2, they delivered very, very short segment of just about 36 bases. And people were trying to piece together a longer genome by assembling these very short, tiny snippets. And what has happened over the years is that for most of these technologies, read length has gone up, while simultaneously the throughput has gone up. Right? And the newest Illumina machines, the HiSeq 4000, and now there's also NovaSeq, which probably sits somewhere here, they can now deliver about 1,000 gigabases in one run with read lengths of the order of 100, 150 base pairs. But there have been some more recent developments to also deliver long reads, because long reads make it much more easy to actually assemble the genome and find the kind of things that Eric was talking about this morning, these rearrangements, gain-loss patterns, and so forth. And the first player in that game was the Pacific Biosciences machine, which is really a big machine. It's about the size of a room, and it weighs a couple of tons. They manage consistently to generate read lengths of about 10 kb, and you'll get a few gigabases, 1 to 5 gigabases per run. A more recent entry into that field was the Oxford Nanopore machine, which is about this size. It's powered through USB. You just plug it into your laptop, and you can you can start to sequence. And what that delivers, again, about a few gigabases per run, with read lengths on the order of 10 KB and longer. Yes? Oh, yes, okay, very good, thank you. So, when you sequence something, what you get out of the machine is not the sequence from the one end of the molecule to the other, right? So you extract the DNA from the organism, purify it, make sure everything else is gone, and then you'll chop it up into little pieces, because the sequences can only read small pieces at a time. And these pieces, that's what's called a read. Right? Yeah, and this is for the shortest one on that graph are sort of about 30. So you just get a little word, right? 30 letters of contiguous sequence, which might come from some random place in the genome. And the longest one here are sort of 10 kilobases. And 10 kilobases would be roughly the length of the genome of the viruses that I'll be talking about. Right, but it's just amazing to me. Now, these are sort of 8 to 10 orders of magnitude that is on this axis, right? And most of that has been covered over the past 20 years. Yes? Run is switch on the sequencer and wait until it's finished. So, no. The run is sort of one sequencing experiment, right? So you prepare your library. So the library is what the pool of DNA bits and pieces is called that you put under the sequencer. Then you let the sequencer go do its thing, and you get a big file in the end. And what is reported here is kind of the size of that file that you get. Convenient that a base has pretty much the same encoding as a byte. So, anything else? So please keep doing that. All right. So what you heard about earlier, so there is one. I think that Luca, you introduced Lenski's experiment slightly. I saw it in your slides, assuming they. Yeah. So in order to get at this dynamical information, right, not having to. So take a snapshot and infer things about the past, actually being able to observe it one generation to the next. Rich Lenski started this heroic effort to evolve sort of 12 E. coli populations that live in these flasks in 1988 and has been doing this ever since. Not him, right? He has now a bunch of people who do it. But when it started, it was probably him. And Michael Desai and colleagues, they have recently, that just came out this summer, have recently sequenced all these lines at a large number of time points. Right? And what you see in these graphs here, it's a little bit too small to really see, but this is the time, right? This is the start at the beginning of the experiment. This is 60,000 generations later. And the squiggly lines that you see are the frequencies of different mutations that occurred during this time. Right? So what a line like this would mean that at some point here, at one particular position in the genome, the state, so you go from 0% A to 100% A around here, right? Or G or C or whatever, right? But this is sort of one example where you can actually see how these populations change, different variants compete, and yeah, the explicit dynamics of the evolutionary process. I don't want to talk about that. It's a terrific experiment and super interesting. But I want to talk about something, another system where we can get similar data, but in an environment that is not sort of a test tube, but something where these viruses, in that case, actually live in and have been doing so for long time. So they're well adapted to that kind of coevolution, where in this case, when you put something in a test tube, you have a lot of things that happen because these bugs aren't quite used to the test tubes that you put them into, and then there's kind of transient adaptation that's going on. So what I want to talk about is the evolution of HIV, so the causative agent of AIDS. And as most of you will know, HIV is a virus, a retrovirus in particular, that has been circulating among all sorts of monkeys and primates for millions and millions of years, and has been transmitted from animals to humans multiple times. And the most significant of these transmissions is from chimpanzees to humans, that happened probably around 150 years ago that has given rise to what is known as HIV-I group M. M is for main, and then there's N for not M and O for other. Not terribly interesting abbreviations, but HIV-I-M accounts for almost all HIV infections on this planet. There's another group, HIV-II, which is more geographically restricted to Western Africa and is probably a transmission from sooty mangabees. But HIV-I-M is by far the dominating group. So this happened around 120, 150 years ago, and more than 100 million people have been infected with HIV ever since. And this virus has, since its transmission, that was a singular event, right, where one very monomorphic populations of HIV was transmitted to a human. And since then, the virus has diversified in a large number of different subtypes, which now differ at about 10, 20% of the genome. And I'll show that in a moment. Which, just to give you a sense of scale, that implies that the HIV-I sequence changes by about 1 per mil per year. Does anyone know sort of how? So each one of us has 2 sets of chromosomes, right? What's sort of the typical difference between them? 1 in a million, 1 in 1,000, 1 in 10. Let me take a poll. So who is for 1 in a million? Hands up. Okay, I estimate that to 10. 1 in 1,000. That's 1 in 10. Nobody's that crazy, right? Okay. So it's about 1 in 1,000, right? So we differ from chimp by about 1 in 100. But 2 human chromosomes are about 1 in 1,000 apart. And it doesn't really matter where we come from for that purpose. So this is to say that HIV changes its genome by about an amount that every year, that sort of every 2 humans differ, and it does in every 10 years it will change as much as sort of the human chimp difference. So it's a very rapidly evolving genome, which allows us to study evolution in great detail very directly. Now, just to give you a bit of an overview of the phylogenetic relationships of HIV, so what you see here is a phylogenetic tree of all sorts of different HIV and monkey SIV variants. And the HIV-I group is this bit here. And the different branches here are the different subtypes. There is then another HIV group here, another HIV-I other group here. HIV-II sits all the way down here. Just to give you a sense of scale, so if we zoomed in to this HIV-I group here, so different subtype, those would look roughly like this, and that time scale here, that is sort of 10% nucleotide difference. Yes, please. Yeah. Oh, these are all trees built on their genomes. Right, so all these different viruses have been sequenced. Right, and there's a sequence, ACGT, and so forth for 10,000 base pairs. So that's about the size of the HIV genome. And then you compare all these different sequences, and you group similar ones together, and less similar ones, and less similar ones. And you get a hierarchical clustering, and that's sort of an approximation of the phylogenetic tree of the virus, how it got transmitted from one person to another. Right, and in that process, over this point here, that was this human-chimp transmission that happened somewhere at the center of the HIV-I-M group. Right? And since then, that virus has been transmitted from one person to the next, and it picked up mutations. Right, and these mutations, they make up the differences between these genomes now. And from these differences, we can reconstruct that tree, that phylogeny, by sort of standard phylogeny reconstruction tools. And the scale here means that from the center of the tree, the tips differ at around about every 10%. Right? Where so I think sort of Eric's E. coli cloud was more like something like this, right? Sort of 1% was your typical difference, right? 1 to 3. So the E. coli that Eric talked about were roughly this. Right, so this HIV since 1900 makes up a sequence space that's about 3 to 5 times as long. So, you know, all this rapid evolution, as I just alluded to, right? Yes. HIV doesn't. Well, this is sort of a difficult. Multifaceted picture what it actually does in the monkeys. In some animals, it's rather well adapted to the host and sort of arranged itself in such a way that it doesn't cause disease. In others, it is actually quite pathogenic. There's no 0-1 answer here. So all of this evolution actually happens within individuals, right? The virus doesn't do anything outside an individual, and the typical course of a disease is roughly like this, where you have here in red the number of viruses that are found in a milliliter of blood of the HIV positive person. And this is, oops, I'm losing the mic. This here is plotted on a split axis, where this is the first couple of weeks, where that person actually feels sick. It has sort of flu-like symptoms of a systemic viral infection. You have a spike in viral load. After that, the virus load drops and continues to hover at around 10 to the 3, 10 to the 4 copies per milliliter for many, many, many years. And in absence of treatment, that individual will at some point have a sufficiently worn out immune system that the person succumbs to opportunistic infections and eventually dies of all sorts of complications. If you do treat that individual, and that luckily is the case for an ever larger number of people these days, viral load will drop to undetectable level, and as far as we can tell, that person will live a normal life, only that he or she has to take the drugs every single day. The blue curve that you see here is actually sort of a health indicator of the immune system. It's the count of CD4 positive T lymphocytes that decline over an HIV infection. At some point when they fall below a certain level, that's when the immune system ultimately collapses and that person is infected by all sorts of things that a healthy would not be infected from. At the cellular level, what actually happens is that the HIV virus, which is depicted here as sort of a little ball with spikes on it, it docks onto specific surface receptors of the cell, and the capsid containing the genome is released into the cytoplasm, transcribed from the RNA. So HIV is an RNA virus, right? The genome is encoded in RNA rather than DNA. But then it has an enzyme called the reverse transcriptase, which makes from that viral RNA a double-stranded DNA, which is integrated into the human genome. And from that place where it is integrated into the genome, this behaves like a normal gene. New virus and protein is made, and the virus sort of bites off the cell again. And the burst size, meaning the number of viruses that can come out of an infected cell, is easily in the thousands. So this is sort of what happens in infection. At infection, you first have this spike of viremia, and then a very long, prolonged chronic infection. Right? And even though this is the people that are suffering from HIV that are HIV positive here are fairly asymptomatic, right? Many people that are HIV positive during this period, they don't even know that they are positive, which contributes to the spread of the disease. But under the hood, there's actually a lot of stuff happening. It's been estimated that 10 to the 8 cells are infected every single day and removed by the immune system. And the virus repeatedly escapes immune recognition. And that is something that I want to talk about a little later. Yes? Oh yeah, so that is actually that's actually because the adaptive immune system does learn how to deal with the virus to some degree. So it it recognizes the virus, it removes some part of it, and lowers the ability of the virus to replicate inside the body. But it doesn't manage to eradicate it completely. And that's basically because the virus is mutating all the time and continuously escaping the immune system. And that's what we'll see in a second. Because what happens in a typical HIV infection, which right at the beginning, the population of viruses in the body is very, very monomorphic, very similar. So if these are the viral genomes here, the black lines, and the red dots are mutations, you see next to no mutations in the first couple of weeks. But then over the months and years, this virus population develops into a very diverse species of many viruses that carry many mutations. And that is sort of exactly that evolution process that I want to talk about in this lecture. Yeah. Yes. That's a good point. So in heterosexual transmission, around 80% of the infections seem to be such that there's one dominating clade, one dominating virus. Then there is sort of some minority, sort of 1 in 5, where you do seem to have multiple founders. But of course, it's a completely different story if you get a blood transfusion that's contaminated, right? Then you get sort of a wholesale load. But the transmission bottleneck of HIV is actually fairly tight, which is also reflected in a fairly low probability of any one exposure leading to an infection. Oh, that's a red dot. That's a series of red dots. It's not this particular mutation right now. Oh yeah, this would be one that has replaced the original variant, right? So that's a fixed mutation, but it doesn't have any particular significance right now. So what, here it has to some degree. So what actually happens during the first couple of months of an HIV infection, that the immune system learns how to deal with the virus, and I don't think anybody talked about immunology here so far, right? So the immune system, the adaptive immune system, has two different branches. One is the cellular immunity and the antibody-mediated immunity. And in the beginning, in particular, it seems to be that the cellular immunity is most important. And what happens in cellular immunity is that every cell in our body carries so-called MHC molecules that present little snippets of protein, just random proteins that are produced by our cells on the surface. So on the surface, there's some sort of like a billboard of little snippets of all proteins that the cell is making. And most of the time, that cell is only producing human proteins because it's healthy. That's fine, right? So T cells would come along and they scan these billboards that other cells present on the outside. They scan this for anomalies, right? And once they see that this cell is producing proteins because it's infected by a virus, right? It's producing viral proteins. These viral proteins will be presented on that surface, on these billboards. The T cell will recognize that and then induce this cell to kill itself because it's excreting certain cytokines. Now, the virus tries to avoid being recognized and presented by that cell as much as possible, and it does that by mutation, right? And if you zoom into one of these MHC molecules together with that peptide, that looks roughly like this. So you can think of it like a hot dog bun, right? Sort of some sort of a groove here, where the green thing. Apologies to people who can't tell those two colors apart, but I didn't make that bigger. The green thing here in the middle, that would be that snippet of protein, that little epitope that is being presented by the MHC molecule. And what the virus is trying to do, as much as possible, to mutate its own proteome so that it does no longer fit into these things, right? So these MHC molecules, these little hot dog buns, they only present a small fraction of all possible protein snippets, because otherwise it wouldn't be discriminatory. And so the virus tries to mutate such that it no longer fits into here. And it's actually very, very good at that. So if you look at the early sequences from like a 7-day-old infection, you typically don't find any frequent mutations. But if you look 3 weeks later, you find that in a few isolated places in the genome, there are some mutations that have spread to very high frequencies. And those high frequencies, those mutations, they tend to be in the parts of the virus proteome that is presented by MHC molecules. Or sort of right outside, there's sort of a bunch of other residues that are important for the processing of these residues. And after 8 weeks, you typically see a handful, and after 6 months, up to a dozen of sites where the entire virus population has changed its genomic makeup to avoid being recognized by the immune system. So that's a little sketch. In reality, this looks a little bit more like that. This is data from a paper from a few years ago, where they had sequences from 2 weeks into infection, where you already see here, this one has completely, in this place, this epitope B, has been completely replaced by some mutant variants. So every red dot here is a mutant relative to the founder, and the green dots are actually synonymous mutations rather than non-synonymous. But at the beginning, you see almost exclusively, sort of at least at high frequency, non-synonymous mutations that are spreading through the population within a couple of weeks or months. And then later in infection, you see that synonymous mutations tend to accumulate here in this part of the genome, while in this part of the genome, non-synonymous mutations accumulate more and more and more. So this now is sort of 2 years down the road. Right? And this process here was studied at a fairly low resolution, right? You have on the order of 10 genomes per time point. It's a very laborious process to get those. And we set out to study this process in much greater detail using these sort of modern sequencing technologies. And this is what we did in collaboration with a virologist at the Karolinska Institute in Stockholm, Jan Albert, who had access to a large collection of plasma samples that were frozen down over the past, since the late 80s in Stockholm. So Stockholm had the policy that every viral load measurement they made, they froze down. So we have pretty complete sample collections from the late 80s to now, and we identified a number of individuals that sort of met certain criteria, and extracted, well, Jan's group, sort of, and one of my grad students, they extracted the HIV RNA from the plasma, and then amplified it into 6 overlapping PCR amplicons, constructed sequencing libraries, and then we sequenced this in Tübingen on a Illumina sequencing machine. And what you then get is sort of what's called a PILAB, where you have all these different sequencing reads, right? These would be these little snippets that the sequencer gave back to us. In this case, it was snippets of about 500 base pairs in length, sort of overlapping 300 base pair segments. So you put them all together to reconstruct the complete genome, which is not too difficult a task in this case because the genome is fairly short and there's no repetitions. And then you can, at each position, you can ask sort of, do we see mutation, right? So here we have 3 A's and then 4 G's. Here you would have 2 C's and a bunch of G's, right? So of every single position in that genome, we can now dig in and count the mutations and count how often we see a mutation next to another mutation, and all these sorts of statistics we can start to look at. Just to give you a little bit of an overview of what sort of the whole data set we have looks like. So this is a phylogenetic tree of all samples that we processed. Each color here is a different individual. And from a given individual, we have samples, say, from early infection, that was January 2000, up until July 2008. And we sequenced each of these samples at a depth of 1,000 to 1 million, really. So every 6 months to a year, we have a sample that we can really dig in very, very deeply. And what you also see, the chimp-human transmission, that probably happened somewhere here in this tree, right? And then there's a lot of evolution that happened until we, at some point, we observe we observe this virus in the first sample from that individual, and then it sort of continues to move on out, right? Like adding rings to a tree, up with every year being sort of another 1 per mil ring diameter, right? So if you now look into these samples in detail, you see all these different mutations that I just alluded to, right? Each of these dots here has the frequency from 1% to 100% on a logarithmic scale. And these would be exactly the relative counts of these guys, right? And this would be a typical sample here. This is only a small little fraction of the genome, because if I showed you 10,000 base pairs, it'd be a little messy. So this is 350 base pairs. It's a protein called P17, where you have at 4 months only 2 mutations at appreciable frequencies and a few at lower frequencies. If you look 5 years later, you see a whole zoo of different things. And after 8 years, actually a large fraction of them have risen all the way to frequency 100%, meaning everybody in the population has them. And the actual trajectories of exactly these same dots that you see up here are down here. The color should also be the same, even though it's sometimes a little difficult to tell apart. But what you see here in thick lines are all these mutations that eventually ended up that at some point crossed, I think, 20% or something like that as the threshold here. And then there's a whole lot of stuff that is just segregating at frequencies of a few percent, which we don't, but never make it to high frequencies. So, and what this, yes. Oh, EDI. EDI is the estimated date of infection, because we don't actually know exactly, right? So actually all these people were selected to have a negative test and a positive test, but often these two are still like a few months apart, and then you're trying to come up with some date of infection just yeah. But for all practical purposes, is the date of infection. So all these, there's a lot of stuff going on, right? And for some of that is going to be exactly this immune escape dynamics that I showed you before. Some of that is just going to be some other random thing that we can't make sense of. Some more is going to be a reversion, and I'll talk about that in a minute. But this graph here very nicely illustrates the immune escape of HIV, in this case, not from T cells, but from antibodies. So what you see in that graph is, and this data from an older paper by Doug Richman, so they took samples from a person over 2 years, every 3 months essentially. And they took both virus samples and plasma samples, and that plasma contains antibodies. And then you test how well does a particular antibody mix from plasma recognize virus. And you do that in all pairwise combinations, right? And here on the diagonal, that is testing virus and antibody from the same sample. How well do they react, right? How well does the immune system recognize that virus? The thing here below the diagonal is testing late virus with early plasma. And you see it's blue, meaning the early antibodies don't recognize the late virus. While above the diagonal is nice and red, meaning the late antibodies always recognize the early virus. So the immune system learns how to recognize that virus, but the virus is always changing, and contemporaneous virus is typically not recognized, right? The immune system is sort of lagging behind by half a year, roughly, right? And much of the dynamics that we observe in the genome is driven by exactly that sort of coevolution between an immune system trying to catch up, chasing the virus ahead of it, and the virus just being a tiny little bit faster, such that, the immune system doesn't recognize to quell the infection. And one thing I actually wanted to show, because this is only a small little fragment of the genome, we have all those data available on a web page, which you see here, right? So these are these individuals that we sequenced, and we can now sort of go into any one of them and look at those those data in great detail. So what we see here, for example, is the genome, right? So down here, so that's the genome coordinate. It's about 9.5 kilobases. These are all the different genes of the virus and different proteins, and these F1 to 6, these are the fragments that we use to amplify it. And on this axis, you see the coverage, right? So how often this particular part of the genome was represented in our sequencing library. And this is an early sample at about 5 months, right? And now we can go from one sample to the next and see how coverage changes here and there, right? Probably a more interesting graph is the one that shows all the mutations that were absorbed in this individual over time, right? And you can again sort of zip through the time. It's kind of too much to see, but if we zoom into one particular gene, which actually happens to be the same gene that we just looked at, you see that over time here you have this dance of mutations that gradually go from low frequency. Right now, all these dots are at the bottom, right? And over time, they move up, reacting a little slowly. But yeah, so this sort of data is available for all the 11 individuals that we sequenced, and you can download it in various different formats if you feel like playing with it. All right, so just a few summary statistics, just to set the stage of what we are trying to explain. So what you see here on this axis is the rate at which the genomes that we fish out of these samples change over time, right? We compare each genome to the beginning and ask how different is it, and do some sort of sliding average rate estimate here along the entire genome. So these are again the different genes, and you see that there are some regions which are super fast, right? They change at about 1% per year. Other regions change at sort of 1 to 2 per mil per year. These here happen to be the regions that code for the proteins that stick outside of the virus, which are constantly seen by the immune system. Those here are the regions that they code for enzymes, the enzyme that integrates the virus genome into the host genome, reverse transcriptase, and so forth. And these here are the structural proteins, which have somewhat intermediate evolutionary rates. And then there's a whole bunch of little things which typically interact with host proteins and downregulate antiviral defenses and so forth. Now, so I think Luca, you talked a little bit of mutation rate, evolutionary rates, and so forth. I saw from your slides, right? How are we supposed to interpret this sort of a statement? So we are looking at divergence from the ancestor, right? So, and what is really happening is, we at least, one way to conceptualize it, that we have some population of some number of individuals, and that number might change, right? That gives rise to the next population by replication and so forth. Sometimes there might be a mutation, right? So this one suddenly has a blue envelope. This goes on and on and on, and at some point we sequence those, right? And we observe a bunch of mutations, and we compare this to the original state in the beginning. So these mutations, they happened, at least if you look at a short enough segment on the genome, be it a single nucleotide, they happened at some point on these branches of the tree. And at some level, the number of mutations that we see in these genomes relative to the ancestor are going to be proportional to the time that has elapsed, right? And this is certainly true for neutral mutations, right? That just serve that don't change the phenotype of the virus in any meaningful way. They would just linearly accumulate by a rate that is this neutral mutation rate times time. But in general, that mutation rate is not equal to the evolutionary rate, because in some regions, there's diversifying selection, which made these bits and pieces that stick out of the virus actually evolve faster than you'd expect, right? And in other regions, there's purifying selection, meaning that most mutations that come in are actually harmful to the virus and hence don't propagate. in which case you would have a behavior that is slower than this, right? And this is exactly what you see. If you look at the divergence, again as a function of time, you see that the envelope proteins evolve fastest, and this here is the non-synonymous divergence, while the enzymes are slowest, while at the synonymous level, here the dashed lines, all of these different genomic regions tend to be extremely similar to a degree that I wouldn't have had guessed. I would have thought there would be much more difference in synonymous, but I'll come back to that. So there is another, so this is divergence, but there's another quantity that you'd probably like to look at, and that is diversity. And in contrast to divergence comparing every sequence to the first sample, diversity compares sequences from the same sample, right? And here you see a somewhat different picture. Non-synonymous diversity tends to saturate fairly quickly. And it saturates at levels that are very low for the structural proteins and the enzymes. Meaning there's very little mutations, non-synonymous mutations that segregate in these regions at any given time. So they just don't go anywhere, or they rapidly go to fixation. Right? That's sort of what's being said. While in the accessory genome, accessory proteins and the envelope, you do have quite a bit of diversity, sort of 1-2%. But it also tends to saturate if at a higher level. Synonymous diversity, however, continues to increase, in particular in the parts of the genome that evolve slowest at the non-synonymous level. Right? There is a perfect anti-correlation here between the rate of non-synonymous evolution and the diversity that you observe at the synonymous sites. And this has a straightforward interpretation in that in these regions where there's not much happening at the non-synonymous level, synonymous mutations just tend to accumulate sort of one by one. Right? There is nothing that would remove diversity that got introduced in previous generations, while in the regions that evolve super rapidly, right, you have a complete turnover of the population because of selection every 2-3 years, which limits synonymous diversity basically at the same level as non-synonymous diversity. If you compare now these divergence rates here to the synonymous ones, right, you see that these two parts of the genome are actually observed. So if we, for now, and there's a big but here, if we for now assume that these synonymous mutations are actually an approximation of our mutual accumulation process, we would conclude that the envelope proteins here and the accessory proteins, that they actually evolve under diversifying selection sort of faster than you'd expect if they were just randomly accumulating mutations, while the rest of the genome is under strong purifying selection. I guess I said all of that. So let's just take this at face value and sort of try to work out from the synonymous mutations, what would the mutation rates, what are the mutation rates that this suggests? And here we actually are a little bit more stringent in that we don't take all synonymous rates, but only all synonymous sites, but synonymous sites that, in addition to that, don't happen to be in any structures, RNA secondary structures that we know of, and those that are globally diverse, so that any unknown structures might also be factored out. But if you do that, you find a pretty nice and linear accumulation of the different types of mutations, right? A to G, G to A, C to T, T to C. These are all the transitions, and then you have transversions, which are much, much less. And here the thing becomes quite noisy, but there still is sort of a clear sort of linear increase here. And from that, we can actually estimate the entire matrix of transitions and transversions in sort of the in vivo situation where the bug lives in, right? Rather than doing it in cell culture and sort of go from a mutation accumulation line in cell culture. As it happens, these rates agree extremely well with measurements in cell culture, which we didn't expect, but since a lot of us as people had claimed that it's very different what you have in vivo, this was sort of a nice confirmation of the cell culture experiments. Now, that was mutation. The virus, yes, sure. Well, 2, 1.2, 0.8, 0.4. But I just... Yeah, so these are per site, so this is corrected for opportunity. In the divergence, so DNDS, here is clearly about 2, right? Yeah. Well, but you know, the ratio of diversities is not DNDS. Yeah, so the divergence is, you know, DNDS is sort of the rate of accumulation of mutations, right? DN and DS. And that's what divergence is, right? With diversity, all that factors in is also the frequency distributions of these mutations. Because you're essentially, you know, diversity, if the frequency is X, right? In diversity, you're averaging this. But in divergence, you're basically just, you're only averaging X, essentially. Right? All right, so that was the mutation part. What about recombination? I've been talking a lot about different regions of the genome behaving differently, right? And that only works if there is recombination. If they were all tied up in one genome without ever sort of recombining, especially all these diversity patterns should be all identical, right? Because if they're all sitting on one tree, there's no way in which the enzymes can continue to accumulate diversity while the envelope part purges it very quickly, right? So how does recombination work in this kind of a bug? So you have here HIV particles, again, sort of a little sketch. And what I didn't tell you so far is that each HIV particle actually contains 2 copies of the genome. So it's effectively a diploid organism, the same way all of us are diploid. And when it makes a cDNA, so a DNA that's to be integrated into the genome, it actually crosses over between these 2 strands about 10 times. And whenever sort of one cell, so if these 2 things are crossing over between are identical, that doesn't do anything, right? But so what one needs to achieve is that these 2 strains that are inside a virus, that these are different. And that happens only if one cell is infected by 2 viruses, then both of these viruses express their genome inside the cell, and there's going to be co-packaging of here the red and the blue variant, which then in the next infection actually results sort of crossed over genomes. Right? Yes? So actually this used to have two red bars here, but it got lost in some copy paste operation. Sorry about that. Yeah, this used to be something like this with 2 reds, I sort of hid the more artistic version of it by sort of sorry about that. Yeah. But the bottom line is, yes, these viruses do recombine, and they do it in a manner pretty similar to animals and plants, in that they cross over to chromosomes. And so that's a more sort of fine-grained level. So if you had a population, say, with, you know, roughly half of the genome having this mutation, some other fraction having this mutation, which are some distance d apart, right, over time, sometimes there will be a crossover generating the variant that contains both of these mutations. And this will be faster the further they are apart and the longer the time frame, right? And we have looked at sort of measures of these kind of non-random associations which decay, and you find sort of patterns like this. So these are these kind of linkage disequilibrium measures, which basically measure deviation of the distribution of mutations at two sites from complete shuffling, right? It's sort of a correlation measure or a co-occurrence measure of two mutations, and you see here how that sort of decays as you move to places that are further and further and further apart on the genome. While the black dot here is a control where this is not supposed to happen because we were afraid that our library preparation and PCR protocol might generate those chimeric reads that look like recombination when they are. So yeah, there is recombination, and that makes in this particular case in HIV, it actually makes it possible that these different regions of the virus help look quite different in the way they evolve. So why would recombination be a good idea? I don't think that has been discussed, so I just want to give it a very, very brief motivation. So say you are in a situation like this, where you have a population where there isn't much going on yet, but there's one good mutation, the red one, and another good mutation, the blue one, and a bad mutation that happens to sit on the same genome as the red one, which is the black one. Now, the ideal outcome would be, of course, to have the blue and the red together. And in absence of recombination, the only way this would work is that you have another, a second occurrence of that red mutation on a separate genetic background, like here. But with recombination, both of these can actually start to grow in frequency. Right? So they go from very rare, so singletons here, to almost half the population each. And once they are at sufficient frequency, it's quite likely that they meet each other in recombination, and you produce a variant that contains those two, which then can quickly grow to fixation, and everybody's happy. Right? So by allowing the sort of parallel amplification of good genetic variants, and then later on combining them into one, that's sort of the way recombination can greatly speed up adaptation and separate the good from the bad. Right? And it's not obvious, but at least as long as you don't have some nasty interactions between these mutations, it's always beneficial. Yeah, but it has to do with the ability to actually to amplify good things while they're rare. For rare things, recombination doesn't matter. Right? Recombination only matters for common things. And this interplay between amplifying good rare things and then recombining them, it's always going to be beneficial for some kind of Jensen's rule sort of argument. Now, the only way you get this to be not beneficial is if you have a lot of positive epistasis between things, that you have co-adapted complexes that you destroy by recombination. So that's when it clearly starts hurting. Yes, so recombination, so it does that, and it actually makes it does a lot of it. So as you see in this graph, effectively coupling between mutation decays over 100 base pairs. So most places in the genome actually do their own thing by and large, without too much interference from very distant places. And this sort of makes things, at least to some degree, easier, because we can try to look at actually individual site and make sense of what happens at individual site, even though there is sort of a huge grain of salt that this should be taken with. But it nevertheless produced some quite useful results, even though I didn't think this would work. But so let's go through it anyway. So we have a high mutation rate, and that mutation rate was on the order, and I don't think I mentioned that number. So that mutation rate was on the order of 2 times 10 to the minus 5 per site and day. Right, multiply that by 300, and you're roughly at this sort of 10 to the minus 3 per, or 10 to the minus 3 to 10 to the minus 3 to 10 to the minus 2 per year, right? So at least order of magnitude, that's okay. But most sites in the viral genome, other than those where it needs to adapt to the immune system, they code for something important, right? And mutating away from them is going to hurt the virus on average, right? So if you now say that being different from a preferred state comes with a cost S, so selection strength against that new variant, and you, for the time being, take this overly simplistic single locus dynamics at face value, you'd have an equation for the frequency at that locus that sort of increases with mutation rate mu and decreases with minus Sx, right? So that's the selection against it, plus a noise term, which I don't specify right now, but it's we'll be trying to average it away. So this would suggest an equilibrium frequency of about the ratio of the mutation rate and the selection coefficient. And if you were able to measure this equilibrium frequency accurately enough, you could turn this around and estimate that fitness coefficient, right? So the problem, of course, is that probably every site has a different S, right? And each single site is very noisy. So how are you ever hoping to even estimate this at a meaningful frequency? So the one thing that we then sort of try to do is just say, is there a meaningful way in which we can separate our genome into categories of strong selection, less, and weak, right? And we did this by taking sort of 10,000 genomes from the HIV data bank, aligning them all, and asking sort of how much variability do we see in different sites, right? And just groups them by this entropy. And that is what you see here. So the divergence, which essentially is just the average of this equation, where it's a linear equation, you can average in every way you want. And it has a solution which is right, sort of and is that correct, Luca? It needs to saturate with, yeah, it's correct, right? It has this solution, and it saturates very rapidly at a low level for the blue guys here, which are the most strongly conserved, and it basically just continues to increase linearly for the most variable sites with that mutation rate that we estimated previously. So at least there seems to be some signal here, and these intermediate categories, they do something between, right? Yeah. Yeah, so they correspond to the different, you have that alignment of all these different HIV sequences, right? And in some places, we have many mutations, in other places we have no mutations, right? And we sort every single site. We sort into like category 1 or 2. This would be probably 6, right? Depending on how much variability we see in them. And each of these colors corresponds to one of these categories. But blue is a category we basically never ever see any variation, and the red ones are the ones where we see all 4 nucleotides at equal proportions, right? So that's basically so this accumulation of diversity within the person actually has a very clear signal, or is mirroring the diversity that you find globally. But then we actually try to, since we have done this in about 100 samples, right? We have sequenced about 100 samples very deeply. So we're a little bit more courageous and asked, can we actually do this for a single site? And at least this S here is large enough, right? So the frequency trajectory decorrelates fast. So this S is sort of the inverse correlation time of the frequency trajectory, right? And if we have about 100 samples, we can, and they are basically decorrelated in between, we can sort of try to put them all together on average. And to make this somewhat better defined, we need to filter them in some way, right? We only take sites where we initially agree with some sort of consensus variant, and sites that we don't think are subject to selection, that are not targeted by the immune system, because that would sort of change the picture, right? So we filter them to a set of sites that we believe are under purifying selection in that individual, and see what we get. And this is then sort of a kind of fitness landscape for every single nucleotide here, which is for clarity just slightly window averaged, where you have the fitness landscape of the synonymous non-synonymous mutations up here as solid lines, again sort of coded by reading frame. So there's different genes that sit in different reading frames. And down here in the dashed, you have the synonymous mutations. And the most obvious thing that you see is, of course, yeah, non-synonymous mutations typically hurt. Synonymous, not so much, right? You do see that there are some regions in synonymous where you have quite well-defined peaks here, or like here and here, or here at the very beginning, or here in this overlap, because that's where two different reading frames actually overlap, right? So the things that are synonymous in one are not synonymous in the other. The same is true here, right? Here is another exon that sits on top of the G41 protein, and that results in this peak here in the synonymous. So at least in the synonymous, we see very clear signatures of a putative fitness landscapes, even if we look at individual sites. And I just wanted to illustrate this a little bit more by zooming in. So that peak here that you've seen in the synonymous landscape, right? If we zoom into this, that is sort of this panel here. And the red line here is fitness average only of sites that don't change the amino acid sequence, so synonymous, while the blue one is everything. And you see here, overlapping with the so-called polypurine tract, which is an essential element for reverse transcription, is that you basically don't see any synonymous variation. You have a similar picture at the central polypurine tract, which is sort of a subleading signal that can do the same thing. There's a few more like splice acceptor sites. So the complex HIV genome needs to make many different proteins, so it's spliced in many different ways. And you have another example here at the very beginning of the GAG gene, where there is one stem of an RNA secondary structure that overlaps this gene. So even though I was very, very skeptical whether one should do this at all, but it does seem to have, at sort of almost single nucleotide resolution, some signal as to what the fitness cost of these sites typically are. Say it again? No, while mutation rate certainly does vary across the genome and with genomic context, we don't think there's a systematic variation of mutation rate here. Oh yeah, but that is sort of after selection, right? So, I've showed that the rate at which divergence happens in different regions differ, and it will differ here primarily, right? This was a region of very low divergence at the non-synonymous level, and this is consistent with almost all non-synonymous mutations being very costly in terms of in terms of mutation, right? If you mutate those, the virus will suffer, and hence it doesn't pick up those mutations to begin with, right? While here in envelope, it's actually not as well conserved, but that's mainly also because the selection pressure on these parts is much, much higher, right? So if you look at the distributions, for example, so you'd have the distribution of non-synonymous mutations here, where about 60% are so deleterious that we actually never see them variable. So this larger 10% fitness cost bin basically means next to no variation. While the synonymous ones, our estimates sort of pile up around 10 to the minus 3, or somewhere between 1% and 1 per mil. And again, sort of here, so we don't observe things for long enough to be able to estimate the actual fitness cost. It's sort of more of a lower bound, upper bound. While then there is some sites where telling apart synonymous and non-synonymous is not all that easy. So here these are synonymous in one gene, but a region that overlaps with another one. And that tends to has a picture that's somewhat halfway in between, partly because these regions also tend to be sort of very flexible in what kind of amino acid sequence they code for. I can skip that. So yeah, the last thing that I wanted to tell you about adaptation in these viruses has to do again with immune escape and more particular reversion. So we've seen that HIV escapes the immune system, escapes the immune systems of their hosts very efficiently. But we've also seen that most mutations are costly in some way or another, right? And all of us, most likely, unless there's identical twins here, will have different HLA genes. HLA is a locus in the human genome that codes for these MHC molecules that present bits and pieces of virus protein to the immune system. And if you have a different set of HLA alleles, then you'll present different parts of the virus, right? And hence, adaptation to the host immune system happens in different parts of the viral genome in different individuals. Now, if most mutations are costly, and the virus has to adapt to every single human de novo, it'll just keep on accumulating these costly mutations. It'll be a pretty crappy virus after a while. So how does it manage to deal with this? Does it find sort of compensatory solutions to all these mutations that it picked up previously? Or is it just going to undo those mutations and go back to the original state after it is transmitted to a new individual? So this is kind of pictured here. So say this was the initial founding sequence. The immune system will kind of drive it up the fitness landscape, well, drive it down the fitness landscape away from some sort of favorite state. And then once, so the question that I'm asking is it going to find other mutations that basically undo this damage and put it back into a new happy place? Or is it just going to hang out here, wait until it's transmitted to another individual and undo the change, right? So that is the question that I want to address. And I've seen this picture before. That is sort of HIV, sort of group M diversity, where there's in the middle, there's some sort of consensus sequence of all of that, that is at least some loose prototype of this preferred sequence that the virus always wants to go back to, if such a sequence existed, right? Now, our data basically is sort of the dynamics of the last couple of millimeters here on that slide, right? The last 10 years of this tree, which in total are probably 150 years. So we observe a sequence close to, so if one of these branches is now this arrow here, right? So we observe the sequence at infection of individual X, and it will have a whole bunch of sites here, these red dots, at which it differs from that consensus sequence, right? And we can now separately, over time, look at mutations where it picks up new ones that it didn't have before, and whether it loses these red dots and kind of becomes more similar to the ancestor, right? So we can look at these two cases separately, and this is what is plotted here, where we again show this divergence from the founder as a function of time, for sites where we initially agree with that consensus. So these are the sites without the red dots, picking up the cyan dots. And you see it's a very, very slow process, right? After 6 years, we've hardly picked up here 1%, right? And the two different lines are just two different ways of defining consensus, whether that consensus is one of these stars, these explosions here at the end, or whether it's a consensus of everything. And it doesn't really make much of a difference. The other two lines, they show the degree of the loss of the red dots, right? The evolution back to the consensus. And it happens at the rate that's about 10 times faster per site. Such that, of course, these red dots, they only make up a tiny fraction, a small fraction of the entire genome. This will be, this is on the order of these 10%. If you compare it to the center of the subtypes, it's more like 4 or 5%. If you compare it to the center of all of it, it's more like 10%. But they end up contributing about 1/3 of all changes within an individual, right? Even though it's only 5 to 10% of the sites, they contribute about 1/3 of the changes, meaning every second mutation actually brings this virus, or every third mutation brings this virus closer to the middle than further away, right? So you can think of this dynamics moving away from that center instead of taking 2 steps forward and 1 step back, right? Along these lines. So that immediately tells you that the rate of evolution that you observe on these long internal branches is probably a huge underestimate of the actual rate of evolution that is going on in individuals. Oh yeah, and this is, we further stratified this, the difference here at the end. We ended up stratifying this by conservation, because you might think it's kind of obvious, right? So the sites where it's different from consensus, those are the sites that are mostly neutral, so you get more mutations, so that's what it is. But if you actually stratify this by the degree of conservation in that global alignment here, you find something like this, where the least conserved sites actually have the smallest difference between reversion and moving away from the consensus, while the most conserved sites have a difference at around about a factor of 1,000. So if you disagree with that consensus sequence at infection, at a site that is among the most conserved 10% of the genome, you have a chance of 50% of mutating back within 5 years. So it's a very strong sort of sequence attractor. And in some way, you can think of the immune system pushing the virus always away from that attractor, but it's sort of the virus population is in sort of some high-dimensional orbit around some attractors, so spiraling out a little bit and eventually coming to a closed orbit, while then sort of course, because of all sorts of other processes, the center, there is epistasis and compensation, and that's sort of top of that, right? This is not a either-or question. Both of these processes contribute, but reversion seems to be a rather strong player in all of this. Yes. This is true for the entire, the question is whether that was for the envelope gene or sort of other parts, but this is true for the entire genome.
Sorry, then it's interesting. Would there be any stratification if I just look at the envelope proteins versus the rest?
Looking only at envelope is actually a little tricky, because envelope, especially the loops, they do evolve a lot by duplication and deletion. So they are not necessarily in this kind of homologous mode of evolution, right, where you only always derive sequence from a parent by point mutation, but you have a lot of underlinable mess in these loops. So yeah, so it's fairly straightforward for 90% of the genome, and it's a big mess for the remaining 10. And most of this is for this 90%. Yeah, so I'm basically done. There's sort of one little thing that I could do on top of it. And that is basically asking what happens to the virus once you start treatment. in an alignment of thousands of HIV sequences sampled across the globe. So the founder is, so you wouldn't, the reversion wouldn't happen if the founder was the most fit, right? Yeah, this putative most fit sequence is the consensus of all of these guys, and what we are observing is just a tiny little bit here on the tips, right? So, that some individual got HIV positive at this point, right? And we are observing the sequence evolution here, and we are asking what fraction of changes takes it back here, makes it more similar to the center of the tree, and what does it makes it less similar to the center of the tree. Yeah, so basically what I'm saying, it is going to be entirely determined by selection, where selection matters most. Right? And as you go to sites that matter less and less and less, you are sort of not fully in that saturated, in the saturation part of that curve. There's sort of an escape reversion dynamics, which is very fast for some parts of the genome. And they will certainly mean that as you go out further and further and further, the time component of this tree becomes less and less and less pronounced, right? And as a matter of fact, and hence why I'm always so qualifying when I talk about when that chimp-human transmission happened, right? People estimate this to be about 120, 150 years ago. But you do the same thing with all SIV sequences. You come up with a common ancestor of SIV and HIV, or all SIVs and HIV in the 1400s. But we know it's millions of years old. So, but here we are sort of at a level, and that would actually make sense if you take this sort of one step forward, two step forward, one step back picture at face value. You'd expect saturation in this radius when this hits about 20%. So the radius here, instead of 10% being about 20%, and then you'd lost it, right? And that's presumably, of course, at some point, since you do have some degree of compensation epistasis and so forth, right? It's not that you can't push this picture all too far. So at some point, there is going to be, this putative optimum is going to change, but in a much, much slower timescale. Chair left, so I can do whatever I want, I suppose, right? Yeah, so we've been discussing a lot. So what all I've been talking about before is stuff that happened prior to therapy, right? So we had these samples at many points during that infection, and in the untreated infection here is indicated by that red bar. And all these people are alive now. And sort of my colleagues in Sweden, they asked them to come back to the doctor's office and give us some blood. And from that blood, we extracted HIV, which is sort of sitting in the genome of the circulating blood cells. Right? That's the reason why they can't stop taking the drug, right? Because the moment they stop taking the drug, one of these bugs is going to wake up again and reseed the infection. So that's why if you're HIV positive, you have to keep taking this drug until the end of your life, because it's not a curative therapy, right? It's a functional therapy, but it's not a curative therapy. So in this case, we have everything that happened up to therapy, and then we have a snapshot of what's circulating in the blood of these people after 5, 10, 15, 18 years. And it's been a big debate as to what happens in between, and how is this reservoir of integrated virus maintained? Is it because there's some pockets in the body where it keeps replicating where the drug doesn't reach to? Or is it just frozen down and that's what it is? And it's the latter. So if you make a phylogenetic tree of a small region of the genome, where you, again, this is the same color coding as before, yellow being early, black being late. The red dots are the RNA, and then you put the blood, the HIV from the blood on top of it, and you see that it perfectly intercalates the RNA tree, which in some cases were sequences from 10 years ago. Right, and this is for 2 of these infections that we have, and we see basically the same picture everywhere. If we quantify this a little bit more by just asking how far away is each and every sequence from the root of the tree here, and plot that against time, you find the following picture. So this is all aligned such that therapy is time 0, therapy start, and divergence, so that the sequence, the reference sequence here is also consensus sequence at therapy start. And you see that prior to therapy, you have a very distinct slope here, meaning these sequences are moving away from the root of the tree at a more or less constant velocity. And after therapy started, it's just damn flat. Right? There's no evidence that there's forward evolution once therapy has started. Right? The distance of these dots here from the root of the tree does not increase with time. And it's the same as the sample that we had right before therapy. Which is to say that, yeah, therapy just freezes it down and keeps a snapshot of the HIV population that circulated in the 2 years prior to therapy start. And incidentally, that also tells us quite a bit about the turnover dynamics and lifetime of T-cell clones. But I think that I'll have to skip. That's it. Yeah, so I hope I've shown you that sort of deep sequencing now allows sort of a very comprehensive characterization of what is going on within infection, and that you can observe really sort of macroscopic evolutionary moves over a few years in just one person. And yeah, that there is sort of at least some approximation of a fitness landscape which sort of characterizes the struggle of the virus with maintaining functional set of proteins and the need to adapt to host immunity. Yeah, so this is shows up a few questions that I hope I can, some of which I can maybe address on Monday. Is sort of we certainly have a complex dynamics of many mutations, and to some degree the recombination seems to uncouple them, but that's probably not the end of the story. We certainly need to find theoretical frameworks that do justice to this sort of complex dynamics. Some more question would be sort of how important are epistatic interactions. I've sort of downplayed this role here because at least much of what we observe we can get away with explaining without epistasis, and so on and so forth. So these are the people that were involved in this work. First and foremost, Fabio Zanini, who was a graduate student at the time in my lab and who's now in Stanford, and sort of my HIV virologist collaborator Jan Albert in Sweden. These are part of Jan's team here. These three. Krista was a sequencing facility head in Tübingen, and Vadim did some of the the fitness landscape estimation work. And with that, happy to take more questions or coffee or whatnot.
What does the therapy actually do?
Therapy, there's multiple different drugs that address, that interfere with different parts of the virus life cycle. So the most prominent drugs are the ones that inhibit reverse transcription from the viral RNA into the DNA. So because that is a very virus-specific enzyme, humans don't have a reverse transcriptase, that can that you can target easily. And they either target the enzyme directly, or they provide nucleoside analogs that terminate the reverse transcription process. Another set of enzymes that, that inhibits a protease that splits the polyprotein into many different, into the functional components. Then there is an enzyme that a drug that inhibits integration. And nowadays there's also integrase inhibitors, entry inhibitors. So they have drugs for all different parts of the life cycle.
Okay, can you go back to the last slides? Before this. Okay, so related to this, but just one slide before. So what are P1 and P8?
This is a patient 1, patient 8.
Okay, so so next slide. So before the start of therapy, I didn't really get what these points mean.
Yeah, so for each of these tips, right? Each tip here, each orange circle or red circle, black circle, corresponds to a sequence that we got from either plasma before therapy or sort of blood after therapy. And what we measured is the horizontal distance of every tip from the root of the tree, which is a measure of its divergence, right? It's sort of it's the expected number of nucleotide changes from here to here. And that's what's plotted here. So it's the mean root to tip distance for every sample relative to the start of therapy.
So what you're saying is the divergence before the start of therapy was increasing,
right?
But once the therapy started, the virus stopped diverging.
Yeah. So there's basically no more replication once there's therapy, but the virus persists as part of T cells, which are long-lived and have sort of a homeostasis mechanism to maintain T cell clones.